What I'm going to be talking about tonight, though, is definitely the first phase of the development of what we hope will be a long-running center or program focused specifically on research translation related to chronic fatigue syndrome and fibromyalgia. And research translation I'm using that as a term to refer to the fact that we are building on research that has grown out of basic animal science, and we are trying now to translate some of these very, very new findings in animal models into something that we hope will help us figure out a little bit more about what is happening in patients who have chronic disorders involving widespread pain and debilitating fatigue, whether you meet the definition, the medical definition for fibromyalgia and chronic fatigue, or whether these are just components of another kind of health problem that you have. So, the way that this began, the University of Utah was very excited about programs where we could move straight from bench-type research involving animals and move the findings there to something that could be maybe breakthroughs for human and clinical work. And so they came up with some funding, which they called the Catalyst Grants. And our research team was fortunate to get some of this seed money. It's not riches, but it was enough to encourage people to get together, scientists who had not worked together before. Now, this may sound interesting. My husband is the director of our Catalyst Grant, and he and I, believe it or not, 30 years and more of marriage, and we had never collaborated on a project before. So the seed money did something right away. But we also were able to bring in some other very good scientists. And we also used that money as seed money to collect a little bit of data so we could then go to NIH and apply for other funds. And we are fortunate in that we are now recipients of a R21 grant for a pilot clinical investigation. I am a principal investigator on that investigation. And it will be a 2-year grant to generate the first translational data, meaning something where we've taken some animal findings and we're now moving to patients, and particularly looking at patients with chronic fatigue syndrome. And that's going to be the project that we talked about, that Roxy talked about, that we're going to be recruiting for. So the key faculty in this new program or new center, it's kind of changing its status, but these are the people who are the big movers in the program right now. And one, as I mentioned, is my husband, Alan Light, who only does basic animal research, but has a great vision for how these things can move to human studies. And another of our colleagues who's been very important is Lisa Cannon Albright, and her more junior colleague, Nikki Camp, and they are doing genetics and familial research, and they're particularly starting with this wonderful resource that we have here, which is called the Utah Population Database. It's all of this generations and generations of data going back in Utah, where they understand who are the family members in these families, and to do familial studies with this group where you can go back and you can look at not just 2 generations, but 3, 4, 5, 6 generations where you have information on who the family members are. It's a fabulous resource for these kinds of studies. We also have an exercise physiologist, Andrea White, who's working with us. And then I consider myself in this area that is called either psychophysiology or behavioral physiology. And one of our other colleagues is Dick Chapman. He is the director of the Pain Research Center at the University of Utah as well. And there's another individual who's part of his program, Akiko Okafuji, who represents the clinical psychology component of our program. And then we're very fortunate to have as medical collaborators Drs. Cindy Bateman and Lee Smith, who are very active members of your group, so they're familiar to you. So I always have this slide in here to say that what we're working with, at least as part of our focus for these research projects, is chronic fatigue syndrome as defined by the Centers for Disease Control. And when I say that, this Centers for Disease Control definition is the best we have to come to grips with this very difficult and multiply presenting problem. So what we talk about when we write our research grants is we talk about patients who report chronic fatigue that severely impairs physical and mental functioning, is not relieved by rest or recovery, sometimes but not always has a sudden onset associated with illness symptoms, traumatic accident, or a stressful life event. And then according to the CDC's vision of this, there are also 8 different little symptoms, specific symptoms, where they're expecting to see 4 or more of these in a given patient. And the key one that we're most interested in the projects that I'm going to be talking about are the fact that symptoms are frequently worse 24 hours after physical exertion. Now, we're also interested in fibromyalgia syndrome, also called chronic widespread pain. And this is characterized by widespread muscle and connective tissue pain. It has to be present in all the body quadrants. There has to be hypersensitivity to pressure at tender points. But the 2 other key components of this, as far as our projects are concerned, are that physical or psychological stress may worsen the pain, and also the fact that there is up to 70% comorbidity with chronic fatigue syndrome. These are very overlapping disorders, and the overlap is probably greater if you aren't quite so fixed on there needing to be exactly 11 tender points. If you allow people with 8 or 9 tender points. So our model that we're working from is one that's called a diathesis stress model. And diathesis just means that there's some kind of predisposition or special vulnerability that makes these people more susceptible. And in many cases, as soon as you say predisposition, people think, well, does that mean genetics? Well, it can mean that you are genetically born with some characteristics that might make you more vulnerable to develop a disorder. Or you may acquire them due to exposures to toxins or to diseases. But there is some kind of underlying vulnerability, and then in our model, certain kinds of stress exposure then can maybe push you over the edge and then make it more likely that you would either develop the disorder or that your symptoms would get worse, or you may even stress may even play a role if you are in remission in inducing a relapse. So this is the model, the general model that we're working from, and this is something that our whole group is sharing as our vision of this. Now, it is a little bit like the blind man and the elephant. Even though we may share this vision, how we come at the problems is quite different in the different projects. Now, one of the reasons that this is such an exciting area to bring new ideas to, new visions to, is partly because, as you know, chronic fatigue syndrome is not very well understood, and it's not especially easy to manage because of that. But even on a far more basic level than this particular health disorder, scientists do not even know what causes fatigue in anybody. In exercise physiology, the definition of fatigue is generally that a muscle that has been active no longer contracts normally. So you've worked it to the point where it's not functioning normally. But we don't define fatigue that way. When we feel it, the sensation that we call fatigue, we know happens long before our muscles start malfunctioning. We can tell, we can sense things before we get to that state. And then, after that state is over, and that muscle has, if you want to say, quote, recovered and can be back functioning normally, we still feel this state that we call fatigue or tiredness. So what's causing that before and after, it's been a mystery. And we're particularly interested in the fact that after exertion, fatigue and muscle soreness is often worse 24 hours later. Not just 10 minutes later or an hour later, but 24 hours later, there seems to be almost a peak time there where you feel considerably worse. You might even feel feverish. You might feel a state of mental lethargy that accompanies this. So that there's a whole body kind of component to the response that doesn't seem specific at all to the muscles. So I'm just going to skip that one. So in terms of the chronic fatigue puzzle then, one set of new pieces of information that have come out, again, in our translational approach, have come out of animal research, have been these findings on what are called acid-sensing receptors, or ASIC receptors. And the full name that's usually used is acid-sensing inward current, and that's why that ASIC acronym comes in. Now, these are receptors that my husband's could talk to you about for days and days on end, and I'm going. But in fact, there's not been a ton of information about them. They were only cloned in recent years, and basically all the work that's been done with them up till now has been in animal models. However, the interesting thing is, based on these animal studies, their function seems to be sensing these states that we think are fatigue and pain. Now, you can't ask animals how fatigued you feel. You can't even ask them if they feel pain. So it's kind of like we think these are states where fatigue and pain may have been induced by metabolic activity. And what are they? Are they actually sensing then? These are receptors that are responding not just to a single kind of signal. The most common thing that people think of when you think of acid sensing, you would think in muscle activity, there might be a buildup of what's called lactic acid. We know that does happen in working muscle. But they are also sensing, or sensitive to, I should say, other components of muscle activity, including ATP, and part of the reason that they're sensitive to these other components is that they are working as a team, if you want to call it that, with other receptors, other very similar receptors, one set of which is called the P2X receptors that specifically responds to ATP. So these are, again, this is all new information. It hasn't been applied to humans at all, let alone to patients with chronic fatigue syndrome. But one of the things that we're very interested in is the fact that these kinds of receptors, both the ASIC receptors and the P2X receptors, are very plastic in terms of how many of them are present. And that means that they will increase or decrease their population in response to various things, one of which is exercise. So, remembering that these ASIC receptors, they work with other molecules like the P2X molecules to sense what are called metabolites. Metabolites are just substances that are in your body that are products of work done by the muscles, metabolic activity. And so in that sense, these metabolites, the ones that are most sensitive to the metabolites we think are sensing fatigue. Now, when the metabolites get to very, very high levels, when the work has been extremely intense, this creates a state where there is possible injury to your muscles and tissues, and that's when pain, the pain responses would be involved. So I want to tell you that this is kind of been a confusing area for me because when we're talking about receptors, I've always thought of receptors as being just the classic kind of neuron, and Al has finally educated me to the point that these molecular receptors are special in the sense that they, on each nerve ending, there will be these molecular receptors that of various different kinds. In other words, it's not just one kind. It can actually, the nerve ending can have ASIC receptor on it, can have a P2X receptor. It can also have another kind that we're interested in, these TRPV receptors that are sensitive to capsaicin, the hot pepper extract, and to heat pain. So they can have all of those together, and they can all be active or inactive together, and then this nerve ending can get a net response from all of these things. And this is something that's only been recently discovered by a scientist who's a colleague of Al's named Ed McCleskey. So the research that Al is doing in these animal models at the University of Utah is focused on the fact that there may be two different classes of these nerve endings that will be sensing these different metabolites. And number one will be neurons that sense metabolites only when muscle damage might occur. And this is what we consider to be most likely a pain and severe fatigue pathway. Then there'll be other neurons that sense metabolites at levels that are near resting levels, or they'll be responding to less intense exercise. These will be ones that tell you that your muscles are working, and this is a fatigue pathway. So this is a picture of some data that Al collected in just like the last 2 or 3 weeks, very, very recent data. Showing that there are these two different populations. Now, every one of these different colored tracings is a different neuron. If I got that right? No. So, okay. And you can see the ones that are grouped at the top under what's called nociceptors, and the ones that we think are responding to pain. What they're doing, if you look at the very bottom on this figure, where there is it says, resting, mild exercise, moderate exercise, heavy exercise, painful exercise, and ischemic exercise. What he has done here is he's changed this bath that he is putting on these neurons so that the metabolites in the bath are consistent with what you would see in your body during these various states. And these metabolites will include lactate, they include pH, which represents the acid-base state, and they include ATP. So, but he's making these states change so that they correspond to these different levels of exercise. And the ones at the top, these special ones that seem to be responding only when you get to the levels of metabolites that you see during painful exercise, they constitute that pathway that's sensitive to pain. But if you look, there's a whole other group of these receptors that start responding even at these levels which would be normal for resting states or for mild exercise through moderate to heavy exercise. So two different groups. So we have then our first really strong evidence that we have pathways sensitive to metabolites, the same kind of metabolites that your body produces during exercise or even maybe at rest, that can be conveying fatigue, and that can be conveying the kind of pain that occurs under extremely high exercise. Okay, so those are the animal studies, or part of the animal studies, but they didn't really involve either stress or exercise. So why do we think that we can look at these kinds of receptors during exercise, and in particular, of course, in humans, which is what we're trying to do. We're trying to translate to humans. So the first reason that we think we can do that is that Al has also done pilot research in mice, where he exercised them by putting them in a hamster ball. Does anybody know what those hamster balls are? They're just those little like wire cages. And he just rolls them around so that they have to be moving on their little feet right and left and so forth. And he exercises them until they go, basically they don't want to do it anymore. They're at their limit. And then he looked at their responses 24 hours later in terms of these kinds of molecular receptors, these acid-sensing receptors, the TRPV receptors that respond to heat pain, and the P2X receptors that respond to ATP. And he found that all of those receptors had been altered by the exercise in these mice. And these alterations that he found were greater in those body locations in the mice where the receptors are most likely to be translating messages of muscle fatigue and soreness. So here's a picture that looks very complicated, but what I want you to see is that the first set of bars on the far left are the ASIC3 receptors, and again these are the acid-sensing ones, and then next to it are the P2X5 receptors that respond to ATP, and next to that are the TRPV receptors that respond to heat. And what you see is, if you look at the dark bar and the light bar, they're showing increases. So the actual numbers of these receptors have increased dramatically 24 hours after the exercise, but particularly in the cervical and lumbar parts of the body, which are where the muscle inputs are more dense. The thoracic part is probably getting more inputs from like the stomach and intestines and so forth. So those are not areas that are necessarily going to be activated during exercise, and those show much lesser increases. But what you're actually seeing 24 hours later is more of these receptors. They aren't just more active, there are actually more of them. Okay, so now I've struggled through the animal research, and and it is a struggle for me because this isn't something that I'm quite as facile with as he is. So I'm going to, any questions on that? That's why I brought him. He will be glad to answer any of your questions on those. So let's talk now about the part that I am more familiar with, and these are the initial human studies. So our initial pilot study in humans is the first, as far as I know, to show that we can even detect these ASIC receptors in human blood. We're using blood. We don't want to go in and take the body parts that they have to do for the animal study. Blood is pretty replaceable, and people mostly don't mind if you take it. So we're looking at these ASIC receptors, but we're also looking at them along with beta-adrenergic receptors. And beta-adrenergic receptors are interesting because in many other studies which I've done in the past, I've looked at the beta-adrenergic system because it is one of the systems that's most responsive to both mental stress and to exercise. And so we're very interested in it for that reason, but we're especially interested in it in these studies because it is one of the components that regulates blood flow to muscles. So beta receptors help you dilate and get more blood to those muscles when they work. And if they're working well, that helps you get rid of the metabolites that are produced by work. So if they're not working well, there won't be sufficient blood flow and the metabolites hang around longer. So that's one of the reasons, at least, that we're interested in that. We're also looking in these studies at certain cytokines, and one of these is interleukin-6, IL-6 it's called. And we're interested in cytokines, and particularly IL-6, because it is an inflammatory, a pro-inflammatory cytokine. That means simply that it tends to be present and actually promotes inflammatory processes, and they can be connected to a number of the symptoms of pain and discomfort, and that sort of almost, you got a fever kind of feeling that you get. So we're interested in those just to see if all of these may be altered at once and in kind of a coordinated fashion in these studies. So our second human pilot study that we did was looking at increases in ASIC3 receptor populations, but in this case we were looking at responses to exercise, not just simply showing can we detect these receptors, but can we detect changes that happen with exercise? And do those changes seem to go along with changes in symptoms? The symptoms of fatigue, the symptoms of muscle soreness, the symptoms of overall body achiness. Okay, so now I'm going to just warn you that to date we have not moved ahead to study patients with chronic fatigue syndrome or fibromyalgia. Our new NIH grant is just starting. That's what's going to allow us to study patients for the first time, and it's one of the reasons that I'm here, hoping that you will help me by either volunteering or talking to someone who might want to volunteer for these studies. But our initial hypothesis is that in at least a subgroup of CFS and FMS patients, we will see dysregulation in the receptor populations involving these ASIC3 and P2X5 receptors. And what we're expecting to see, this pathway is intended to be protective. It's intended to allow you to sense when your muscles are getting perhaps overworked. And but what we think may be happening is that this pathway, instead of being protective, is now too active. It may be responding inappropriately when there's no risk that you are going to be overworking your muscles, yet you might feel like you are. So our hypotheses are that these patients may have high levels of these receptors even before they exercise, and or the patients will increase the populations of these receptors to a greater extent, or maintain these increases for a much longer duration after they exercise. And all of these things may be true. Okay, so we will at the same time, because this has never been studied before, we will actually be studying whether fitness level, whether highly fit versus less fit individuals respond differently in terms of these receptors after they've done a fatiguing physical exercise. And we'll be studying whether men and women differ. Again, this is so new that nobody knows the answers to these questions. Okay, and I'll just... Here is the actual protocol that we're going to be using. It's really a simple protocol. We're asking volunteers to come in. They have a rest period when they're just sitting. Then they have a period of moderate bicycle exercise on a stationary bike for 25 minutes. We're trying to set the exercise level to be only moderate, and my colleague, Andrea White, who is the exercise physiologist, has set up the protocol so that we'll be using heart rate as one of our cues to keep the exercise just in the moderate range. And we'll also be repeatedly asking people to rate how strenuous they feel the exercise is in order to keep it in the moderate range. So we're not trying, this is nothing like maximal exercise or exercising people to exhaustion. And then this is followed just by another rest period of 30 minutes. Now, the tricky part in the study, if you want to call it that, are those B's that you see. Those stand for blood samples. We do need people to be tolerant of needles because in order to get measures of these ASIC receptors and the P2X receptors, we do have to draw blood 5 different times in the protocol. Once before the exercise, once 30 minutes after the exercise, and then again 8 hours, 24 hours, and 48 hours after exercise. And at the same time that we take the blood draws, we will get people to give us ratings of their current feelings of how fatigued they are, both mental fatigue and physical fatigue, and also we get ratings of pain. So, what have we done so far? Well, at this point, we have only collected data on 4 healthy volunteers, 2 men and 2 women. And I have with me the example data from the first female volunteer right here. And I'll show you the rise and fall in populations of these ASIC receptors and also of these other interesting receptors, the beta receptors, the beta adrenergic receptors. On my leukocytes, that's just a blood cell, during the 48 hours after the moderate exercise. And I will tell you that even though I exercise quite a lot as a rule, I don't bicycle. So that bicycle exercise for me actually makes me a little sore. So here's a picture of my data. And what you see is the beta receptors are in the open, the white bars, and the ASIC3 receptors are in the darker bars, the gray bars. What you have first here on the left are the levels that were existing before exercise when I was just resting, and then you look at immediately next to that are the receptors 30 minutes after exercise. And basically there isn't any change immediately after exercise. There's the same population size essentially of these receptors 30 minutes after exercise. So if you were only looking at the short term, you'd say nothing is happening. But then when you get, I did 6 hours. We aren't asking our volunteers to anymore do 6 hours, but I did a sample at 6 hours as well. Now, at that point, you're starting to see some increases. But by 8 hours after exercise, look how the populations of these receptors have grown. I mean, it's really extremely dramatic. There's just many, many more of these receptors around in my blood. And that is maintained, in my case, at the 24-hour period. And again, this is, at 24 hours is when I, felt achy and fatigued and, wanted to take the elevator instead of climbing the stairs, that kind of thing. But by 48 hours, at this point, mine have returned almost back to the pre-exercise level, still a little bit high. But now, what we are hoping to find, perhaps, is that at least some of our patient volunteers, when we test them, we may see that even at pre-exercise, they may start at a much higher level than I did, and they may actually show changes earlier, or my strongest belief is that when we look at the 48-hour sample, we're not going to necessarily see that they have returned back to their pre-exercise levels. That's my true belief of what's going to happen. Okay, so this is what we're needing to learn next. We need to find out what happens to these molecular receptors in chronic fatigue and fibromyalgia patients. And one of the other questions that is really interesting for us down the road, we know that there's a genetic basis for these receptors, that they have animal models that completely lack ASIC receptors. For example, one of Al's colleagues, again, is doing research with that particular genetic model. So we know that these things are genetically determined, and we are hoping that down the road, in collaboration with our geneticist colleagues, that we can maybe pursue this actually out to see if there are genes that may involve these molecules, whether it makes it less likely that they downregulate, or may simply mean that they are overexpressed to begin with. whether or not that has any connection to the disorder. So we can actually hope that we'll get to that level as well eventually. Right now, the familial studies that are going on, directed by Lisa Cannon Albright, again, are using not specific volunteers, but are using the Utah Population Database and linking that up with medical records. And so what she has done, she's been looking to see if there is a familial component to chronic fatigue syndrome seen in those patients who are part of the Utah Population Database. And her concept is that, and she's used this concept very successfully with breast cancer. Her work, she really has done some of the best work that's been done looking at and eventually leading to identification of specific breast cancer genes. So she started with this same approach in that case. So she's looking for any disease with a heritable component, and she then checks to test her hypothesis that that should occur in a higher frequency among relatives of cases, meaning individuals who who have a specific diagnosis in the medical database, than you would see in random controls. So what she did in the Utah Population Database, she looked specifically for the code, the ICD-9 code for chronic fatigue syndrome. And even though that's a fairly recent code, it didn't even exist a number of years ago, she was able to find in the Utah Population Database over 550 cases who were diagnosed. And she was able then to tag that to their relatives, and they had over almost 3,700 total relatives. And what she found is that she observed many more cases in the first-degree relatives and even in the second-degree relatives of the original cases than she would have expected. And so she would have expected to get, I guess it's on the level of 8 times, I guess, for the first-degree relatives, and just 2.5 times more in the second-degree relatives. Now, first-degree relatives, they've been studied a lot, and there are other papers showing this familial pattern. But second-degree relatives have not been studied that much, and she actually had further data that went out to third- and fourth-degree relatives. And that's where she says there's a much more compelling case for a genetic component, if you can carry it out past the first-degree relatives. And why is that? It's because first-degree relatives tend to share a great deal of the same environment as well as genetics, and it's very hard then to pull that out. But once you get to second- and third-degree relatives and so forth, you don't have as much in common with your second cousins as you do with your brothers and sisters in terms of shared environment. So at that point, it really strongly supports a genetic component. Okay, well, this is our model for all the factors that we are looking at in our studies of fibromyalgia and chronic fatigue syndrome. And I'm just going to go ahead and say that this is in part built on work that I did at the University of North Carolina with patients with chronic fatigue, fibromyalgia, or temporomandibular disorder, that's the TMD. And in this study that I completed previously, I had already previously seen dysregulation in factors that were involved with the regulation of blood flow. And also I saw differences in these cytokines. And I'm just going to kind of speed ahead and show you that this one particular figure where we were looking at norepinephrine, which is one of the major neurotransmitters, and it's one in particular that may be connected to the way your whole cardiovascular system responds. And so what we did in this study, these are just comparing the chronic fatigue patients that we studied versus the healthy controls. And we found that the chronic fatigue patients, not only did they have deficient levels of norepinephrine to begin with, they also showed less of an increase when we made them do certain things. like rise to stand. So it was a sign that their sympathetic nervous system was dysregulated. So we've already got one kind of dysregulation going on here. And in this same study, we used a drug called propranolol. Oops, sorry. Going back that way. We used a drug called propranolol, which blocks the beta-adrenergic receptors that are half of the sympathetic nervous system. And what we found is when we did that, we were able to normalize, for the most part, the responses of these patients. They looked much more like the controls once they'd been given this beta-adrenergic blocker. So that was one reason we decided to really focus on beta receptors as part of our future work. The other factor that we're keeping in our current studies are these pro-inflammatory cytokines, and in particular interleukin-6. And it's because in this previous study that we completed, we saw that both chronic fatigue and fibromyalgia patients had elevated levels of interleukin-6 in this protocol. And we were also able to show that propranolol, this beta blocker drug, reduced pain in these patients. If you look at these pain ratings that people gave us during a placebo session and the ones right next to it that were taken at the same time point after they'd been given this beta blocking drug, we were able to reduce their pain levels by about 30% with this propranolol. But one of the interesting characteristics was the patients who showed us reductions in pain were the same patients who showed us reductions in their interleukin-6 levels. So if they didn't show us a drop in interleukin-6, their pain levels stayed right where they were. So this suggested to us that the beta receptors and beta-adrenergic activity are tied to the cytokines, and that all of that has to do with some of the symptoms that these people are experiencing. And we feel that in our current study we're going to be able to see some of the same connections and connect it on further to the molecular receptors that I've been telling you about. Okay, so we need volunteers with chronic fatigue syndrome, and we need particularly people who are willing to go off medications with medical approval, except for antidepressants, which they can continue taking. So we need people to stop taking them just for a short period, just for 48 hours before the exercise and for 48 hours after the exercise. So it's a total of 4 days that we would need people to interrupt their routine medications. And again, this has to be with approval of their physician. And we need people who can just make a 90-minute visit one morning and then 2 20-minute visits on the next 2 mornings to the hospital so that we can do the exercise testing and do the subsequent blood draws. And we need people who can tolerate 25 minutes of moderate cycle exercise and can tolerate meals.
Do you also want fibromyalgia, or do you want only chronic fatigue?
We want both, and in fact, our goal eventually, if we accomplish it, is we will hope to have a large number of patients who have just one diagnosis, and then we also want a large number of patients who have, as I say, comorbid, that they meet criteria for both. And part of the reason we're interested in that is because we think that there's not going to be every patient will show us the same pattern. So we're really trying to zero in on what characteristics of a patient may predict a certain pattern of differences in these receptors that we will pick up on.
What is the difference between chronic fatigue and fibromyalgia?
Okay, the question was, what are the differences between chronic fatigue and fibromyalgia? Let's see if I can get back here. And
while Dr. Light's looking at that, can I just ask people, please, when you have a question, raise your hand, And let me bring the mic to you first so we can get your question on the tape.
So chronic fatigue syndrome, usually the key factor in chronic fatigue syndrome is that the primary symptom is fatigue. Pain sometimes is a component, but it's not universally a component. The fatigue that is not relieved by rest or recovery, and all of these other symptoms that I've listed, these 8 symptoms at the bottom, several of them involve pain: a sore throat, swollen glands, muscle pain, joint pain, headaches. Those are sometimes part of it, but they're not required. You don't have to have any or all of these. You could have only symptoms that were of a non-pain character, and you would still meet the criteria for chronic fatigue. That's, as I say, it's much more difficult in a sense to define chronic fatigue syndrome, whereas defining fibromyalgia is somewhat more specific, but in some ways a little more arbitrary too because of that. So fibromyalgia is defined by very widespread pain that you feel in all the quadrants of your body, but it's generally heaviest in sort of the muscle joint regions. And at the same time that you report that this pain is debilitating, even when you're not touched by anything, you're also hypersensitive to pressure at certain tender points. And there are 18 of them, so that somebody can sit down and actually press on you these 18 points. And if you say ouch to 11 of them, you would meet criteria for fibromyalgia. Yes?
I was wondering if you have any kind of age limits on this study, and also, do you care if the people in the study, if their extended family is not from Utah, so that there wouldn't be any linking to the Utah database?
Oh. The 2 studies are quite independent. So the study with the Utah population database, it's going on anyway, and no, it's not at all necessary for the volunteers for my study to be part of that. Now, the age limit, we're trying at this point to keep it to the premenopausal ages for women, at least in this initial study. I don't plan on that being the case in the future, but
so is that defined by if they've gone through menopause, or are you using a specific age?
Well, we're right now we're kind of aiming to be 18 to 49, but it is intended to be premenopausal. So some women go through menopause before and some after that. So that's just sort of the guideline. It's around 49.
I had a serious episode of this when I was younger and had small children. For almost a year, I was, well, you could say bedridden, except I couldn't be. So I crawled to the kitchen and fixed food. That's how it was for that year. And then I got better. But 5 of my 6 daughters have chronic fatigue to some extent. One does not have it at all. She doesn't think it's a real illness. And I wondered if I could ask you a question that's off of the study. Do you mind?
You can try me.
Okay. I wonder if you've studied the effect of oxygen on this illness, on the fatigue illness.
I have not. Al, have there not been hyperbaric chamber studies that were done? Yes. A physician from the Medical College of New Jersey, who is one of the consultants on our project, Dr. Ben Nadelson, has done some studies with hyperbaric chambers. And my recollection about those studies is that there was a subgroup of patients for whom this was a benefit, but not that it was a universal response.
Well, several of us are dramatically, excuse me, are dramatically less fatigued at a lower elevation. Dramatically. And in one case that I saw, I just always felt all these 100 years that oxygen plays a huge role in this illness. So there's something going on with the oxygen.
Right. And, you know, in some ways that is consistent with our notion about blood flow being important, because, again, that's what carries the oxygen, is the blood. So I certainly see that as a piece of this puzzle. And hopefully, again, as we find out a little bit more about how these metabolic components work, that oxygen will end up being a piece of that whole puzzle that we'll also be putting in. And again, moving animal studies are in some ways easier to sit down and look at, well, what will oxygen be important as part of the whole metabolic mix, because in those studies then you can control all of the other metabolic factors and just manipulate the oxygen. And that's part of the reason that I think our center has a wonderful chance of really answering questions, because when you make a suggestion like that and he's listening, it can start at that level where we can really get a good look at how these factors are all working together in the animal model, and then moving on up to the human model. Thanks. So yes, there's someone who's been waiting.
I just wanted to make a comment that I am on oxygen. I sleep with it and I wear it during the day as well. And my doctor has noticed that mentally I am much sharper, and if I wear it, it really does help reduce some of the pains that are chronic, and it takes away a lot of the brain fog. And that was something that she just saw immediately. And then one other statement, I spend a fair amount of time just above sea level, and that does give me a lot more relief.
This is all very helpful. I'm really glad I brought the instant translator to research studies along with me.
Are there any other questions?
If you have chronic fatigue, fibromyalgia, and you meet your age criteria, but you have another illness as well, can you participate or not?
Yes, you can, providing that the other illness isn't one that makes you at greater risk during the exercise. For example, we don't want to test patients who have cardiac arrhythmias or who have, we can't test people who have anemia. Again, that's because of the blood draws. But those are the only kinds of exclusions, ones that might make it unsafe for you or put you at greater risk if you participate.
Could I ask a question? I noticed in your study you're going to be doing a blood draw during rest while being seated, and I wondered if you had ever considered doing that after resting lying down.
Actually, we're doing this in one of these standard shock chairs. So when I say seated, you're kind of in a semi-reclined position, and it's simply we do it that way because just in case you get a little lightheaded, it's really easy then to push you completely flat. But part of the reason we didn't want people to be in an actual reclined position to begin with is there are some postural effects. So we don't want people to be changing posture. In fact, chronic fatigue patients are really very sensitive in many cases to moving from a fully reclined to an upright posture. We're going to have you, after we take that sample, move over and sit on the bike. So we want you to be more or less in the same posture. We don't want you to be completely changing, because otherwise we'd be eliciting some other responses during the exercise task.
Does anybody else have any other questions? Okay.
Have they made any study on the high volume of using a high amount of a Gauss towards magnetism in helping to alleviate pain and also to help bring about more energy? So I have found that it's been phenomenal in changing my life. It's really helped me a lot with my fibromyalgia and chronic fatigue.
I know, this isn't one of the components that we're studying, but I remember that it was included in one of the reviews that was presented to our group not that long ago. It's certainly, it's one of these things where I think everybody's thinking ahead to treatments, and I don't blame you for wanting to go there, because that's where the real answer is as far as you're concerned as patients. But at this point in time, I'm afraid that our program is still pretty much focused on finding out what might be going wrong in the system. So we're still, I guess, at the beginning baby steps. And we won't really be at the level of comparing different treatments probably for a number of more years. So.
And Dr. Smith, you wanted to say something?
No. well, let me just comment. I did do a study using a thing called a transcranial electrical stimulator that uses energy, and there was a subgroup that did statistically respond to that.
I have chronic fatigue syndrome, and I went to visit my daughter. She lives out of state, and I was with her for about 6 weeks. And after I came home, she said that she started having symptoms of, and she has fibromyalgia now. And I wondered if there's any way I could have given it to her. My other children are afraid they're going to get it now.
Well, my strongest belief would be that if you gave it to her, you gave it to her at birth. It's part of the genetic package, but it's very possible that one thing that does happen is that when people have been associating with each other, they recently reported that, for example, married couples who are completely unrelated genetically are more likely than random people to share this diagnosis. And part of the reason there is shared environment. So some of the things, you may have actually a very common environment also with your daughter. And another factor may be simply, that they pointed out, is that simply people are more alert than to the specific kinds of symptoms involved. So instead of going to the doctor and saying, I have this peculiar pain in my hip or something like that, they go in and they say, Doctor, I have pains in a lot of places, and I wonder, can it be fibromyalgia? Because I know about fibromyalgia. So some of it is just that.
It's not my fault.
How common is it for someone that has chronic fatigue to have relapses?
Very common. I think the statistic that I saw reported by the Centers for Disease Control were that the majority, I think it was close to 60%, report that it has returned multiple times. So I think that was 60%. So relapses are, more people will experience them than don't.
My question deals with, back to I guess more dealing with your hypothesis with the ASIC receptors and in dealing with the treatment. I guess what, have you guys thought that far? You didn't really speak to this, but have you thought that far out?
Well, Al was just talking about that. Al, why don't you take the microphone from him for a second and say what you were...
Yeah, the fact is we know nothing about what controls the level of these receptors. We don't know why they go up. We can say, okay, exercise takes them up, but that begs a question of what specifically in the body is causing them to go up. And we know even less about what makes them go back down again. There are a number of mechanisms by which we know this can occur, but we don't know if it is occurring. There are a number of substances called transcription factors. These are molecules that cause more DNA to be transferred and made into messenger RNA, which makes more protein. Exactly what transcription factors are involved here, we don't know. We know that at least one of them is one factor is a factor that comes out during inflammatory processes. That's one of the factors that causes the ACE receptors to go up. We don't know what makes them go down. So as a result, we don't really have any idea what kind of treatments would make them go down. The best guess that I would make right now is what makes them go back down is your blood flow increases in your muscles. Now, if your blood flow increases in your muscles following exercise, partly because of the reflexes that these particular receptors activate. So these receptors themselves are activating reflexes that increase the blood flow, take away the metabolites, and therefore the receptors can go back down again. Another thing that increases the blood flow is growth of your blood vessels. You all may know that when you exercise, one of the things that happens to your muscles is they get stronger and the blood supply to them increases. As that blood supply increases, it's increasing because blood vessels are growing. If you have more blood supply in your muscles, that may turn down the amount of metabolites that the receptors are seeing, so they go down. That's all I know right now. I don't know anything more specific. As a result, I really can't tell you what kind of treatments would work. That's part of the reason we're doing this study. Once we I could make up anything I want, but it could be 180 degrees off. I could tell you something would make you worse. So, sorry.
Could I ask another question? I'm kind of hogging the mic here since I have to. You did some kind of study, or you're going to do some kind of study with an MRI measurement. Is that still going on, and what happened with that?
Actually, yes. I kind of tried to keep to the study that we're most actively recruiting for right now, but we are very interested also in the characteristics involved in the central nervous system processing of fatigue. And we're interested in how that may be different in patients who have chronic fatigue versus individuals who are healthy. So we are doing studies at the university where we are using functional magnetic resonance imaging, fMRI, and these studies are involving testing people in the scanner, in the magnet. We have them do one of the tasks. It's a very simple task. It's just finger flexion, very simple, very small movement. And we have people do that, and we're doing imaging of their brains, the activity involved in their brains during just a simple motion like that before and after they've made their hand very fatigued by squeezing very hard on a tube, a fluid-filled tube. So if you squeeze for long enough, time goes on, it gets tireder and tireder and tireder, and then you release it and then try to make that motion. It feels very different. It does to me, anyway. And we're assuming that the way this information is going to be represented in terms of your brain is going to look different too. So it's actually quite a simple study. So that's one fMRI study that we currently have in progress. We're also looking at patients with multiple sclerosis in that particular study. So we actually have chronic fatigue. Fatigue is a very common characteristic of patients with MS as well. So we're looking at them. And then the second study, we're going to be trying to look at the characteristic of mental fatigue rather than physical fatigue. So we're doing a similar protocol where we have people do a little mental task, and it's a very simple mental task to begin with, just some very simple addition tasks involving just one-digit numbers, like adding 1 and 9 together. It's a very simple task, and then we're inducing mental fatigue by having them do a very complicated task for a few minutes, so that your brain's kind of a little bit tired afterwards, and then we have them go back and do the very simple mental arithmetic task again afterwards and do imaging during that. So we're looking at both physical fatigue and mental fatigue, and we're looking for changes in the, these are actually looking, fMRI looks at blood flow too. It's interesting. This is another way of looking at blood flow, but it's looking at blood flow to the brain specifically. So that's those two studies. We'll be recruiting for those as well. I didn't bring any flyers for those, however.
If you have a sheet of paper, you want to take names and numbers of people who are interested, what are the criteria for those?
Basically, in that it's a similar set of criteria. We're going to ask people to go off their medications again, except antidepressants, for 48 hours before participating. So that part's the same. The difference is that since it's just a one-session study, there's no needles involved. You just then you can go right back on your meds after the 48 hours as soon as you're done with your fMRI study. So very much the same population.
Do you have to be premenopausal
Actually,
for that
no,
too?
not for that one. You're right. Postmenopausal women may participate in that one.
I had one other question that someone asked about relapses. I wonder if anyone really ever recovered from chronic fatigue. It's been, day after tomorrow, it'll be 40 years since I first hit the chronic fatigue, since I first got it, and I've never had one day the same since. I wonder if anybody really ever recovers.
Lee, I don't know if you have a...
The answer is in some types, the fatigue can go away and stay away with some treatment. It all depends on what's causing the fatigue. There's sort of chronic fatigue syndrome and then chronic fatigue syndrome, and you have to subtype that, and you can't always tell which type you have. But the answer is, some people, yes, it goes away.
Sorry, this is kind of off of his, and I know. So is that the same for fibromyalgia?
Yes, of course.
My question is, some people seem to, I wonder how much hormones are involved. Some people tend to improve when they're pregnant, or after a pregnancy there's more problems, menopausal problems that seem to aggravate or actually improve the condition. How much do hormones have to do with this?
Well, there's various theories about how important they are. Again, one of the issues that you were bringing up is the same thing that Lee was mentioning, that there are subtypes and subgroups, and since for some people it makes symptoms worse, for others it makes symptoms better when you're in a high estrogen state like pregnancy, or if you take birth control pills. But because it can go either way, it kind of almost, there's so much confusion that's created by that kind of observation that it can't be verified. But one of the things that is obvious is that there is a higher preponderance of women sufferers than men. So perhaps what we may be seeing is something that's operating at a level where estrogen is part of gender, and then at least at that level, there's almost certainly some connection. But again, the only thing I can state specifically is that that's something that everybody has in mind. Every model has that in mind because of the gender differential, and there are ways in which various hormonal states do affect beta-adrenergic receptor responsiveness. There's a literature on that. We are looking at gender in terms of our study when we're looking at these exercise responses. So we're going to specifically be looking at whether there are differences between men and women in the way these ASIC and other molecular receptors respond. So we hope we'll be able to indirectly tap into some of the answers to these questions.
Well, I have all three of them, the chronic fatigue, the irritable bowel syndrome, the fibromyalgia, and now I have sleep apnea. Does that go with it?
I didn't think so, but does anybody, it does? I mean, various sleep disorders do, but I didn't know sleep apnea specifically was one of them.
About a fifth of fibromyalgia patients have sleep apnea, and when you treat it, it improves.
Dr. Smith just said about a fifth of fibromyalgia patients have sleep apnea, and fibromyalgia improves with sleep apnea treatment.
Right, if you have it.
If you have it. Of course, if you don't have it, that wouldn't help. Well, it's time to wrap it up, I guess, and I want to thank Dr. Kathleen White very much for coming to talk with us tonight.